Hi, I'm Jared, and in this short video I'm going to give you a quick overview of the liquid handling tasks available in the ePrep software. The tasks can essentially be categorized into three main groups. The most common group is the one-to-many. The tasks in this group take an aliquot from one input container and transfer the liquid to many output containers, hence the name one-to-many. The second group is the one-to-one. This involves taking an aliquot from one container and transferring it to one only output container. The third group is many-to-many. This involves taking aliquots from multiple input containers and transferring the liquid to multiple output containers. We'll have a look at a deck display of the ePrep to explain this more fully. So let's get started. A good example of the one-to-many task is the Add Diluent task. In this example here, a defined volume is transferred from a diluent container to each of the output vials. The Makeup to Volume task looks very similar to the Add Diluent task, but in this task, the final volume is defined and the software will calculate for each vial how much volume needs to be dispensed to reach this final volume. Other one-to-many tasks include the Add Internal Standard task, as seen here, also the Add Reagent and Add Reagent and React tasks, and Add Surrogate are all considered part of the one-to-many type. Two further tasks, which are one-to-many, are the Serial Dispense task and the Variable Dispense task. These two tasks are very useful for the preparation of a series of calibration standards from a stock standard solution. The Serial Dispense task can be used to transfer a stock standard to a series of autosampler vials in increasing defined volumes. The Variable Dispense task will also transfer a stock standard to a series of autosampler vials, but with an increasing increment volume, which could be different each time. An example of the one-to-one task is the aliquot sample task, where a defined volume is transferred from an input to an output vial for as many input and output vials that are defined with the task. The serial dilution task can also be included as a one-to-one task. For this task, an aliquot is transferred from a first container, which could be the stock standard concentrate, to another container. In this example, this is an autosampler vial. Then an aliquot is successively transferred from one container to the next, resulting in a serial dilution. This task can also be used to serial dilute a concentrated sample to bring it within the range of the working calibration standards. The final task is the many-to-many task, where aliquots from multiple input containers are transferred to each vial in an output group. An example of this could be three different internal standard solutions which are all added to each autosampler vial. A summary of all the liquid handling tasks can be seen here in this table. Hopefully, when creating your workflow, you will now be able to see which of these liquid handling tasks is suitable for your particular application. Thanks for giving me your time, and I'll see you next time.